everyone. I welcome you all to the class of Water Supply and Wastewater Engineering. I am Dr. Devaanshi Jagwani, Associate Professor in the Department of Civil Engineering at IPS Academy, Institute of Engineering and Science, Indore. So, as we have seen in the previous section, that water is required for different purposes. But what is important that we should know the quality of water which is required for our essential activities. There are certain impurities in water that comes in the form of physical, chemical and bacteriological aspects. So it is necessary to assess the water quality. To assess water quality, we need to go for different types of characterization and analysis, like the physical, chemical and bacteriological analysis of water. There are different reasons for the impurities that come into the water. Whenever water comes in contact with mineral and organic constituents, which is in the form of upper regions of the earth's crust, that constitutes the mineral impurities into the water bodies. Three principal sources are there in the form of groundwater, upland surface water, and lowland surface water. Underground waters are mostly available in the form of deep wells, boreholes, or springs, which are basically rich in mineral content, high total dissolved solids, and hardness. The organic content is however low into the groundwater sources because the natural soil acts as a filter, which is in the form of a rock strata. The waters are taken from upland streams and lakes and that usually have less mineral content and low TDS and hardness levels. However, they are rich into the organic matter, especially humic and fulvic acids, which tend to give them a yellowish tinge. Surface water bodies is derived from lowland surface sources and they sometimes originate in the form of springs. So the same general characteristics, they have similar general characteristics as that of groundwater. River and canal waters, especially in industrial areas, they tend to accumulate man-made pollutants, so which it should be recycled and properly treated before disposal. All the sources of water can be broadly classified into surface resources, which is constituting the streams, rivers, ponds, lakes, and impounding reservoirs, and the subsurface sources, which is in the form of underground waters. Whatever impurities are coming into the water bodies is classified into four categories, that is dissolved substances in the form of inorganic salts and organic matter. Inorganic salts specifically like the calcium, magnesium, chloride, nitrates and sulfates. Organic matter in the form of humic, fulvic acids, pesticides, detergents, etc. Suspended matter is further classified into three types: colloidal, inorganic and organic. Colloidal matter constitutes the clay peat acids. Inorganic, which is in the form of sand particles. Organic, which is in the form of industrial, domestic byproducts. Living matter consisting of the micro and macroorganisms. Microorganisms like the bacteria, algae, viruses, and etc. Macroorganisms, basically the living organisms like the fish, shrimps, worms, larvae, and so on. Gases, which is in the form of oxygen, carbon dioxide, sulfur oxides, nitrogen oxides, ammonia, etc. So basically the impurities that are present in the water is classified into four different types: the dissolved impurities, suspended, living matter, and gases. Potable water is the water which is drinkable, which we can consume directly. That sort of water is called as potable water, also known as the wholesome water. Mostly it is colorless, odorless, has a good taste, and it is free from all harmful mineral substances. As we have seen in the previous lecture, for water supply we need to have a water source. So we need to have a water source from where water is withdrawn. Then it is taken to the treatment area with the help of a pump if required. This treatment area treats the water, brings it into a form which can be used for drinking purposes directly. Then it is taken to a reservoir from where it is supplied to individual homes which cater to the requirements of the consumers. So sources can be either surface water or groundwater. Most commonly surface water is preferred, and if there is a river, river is taken as a primitive supply for water. The water quality is tested for various objectives. First, it can be used for checking whether the drinking water is safe or not. Second, it can be used to identify problems into it. Third, it can be used for precautionary measures. Fourth, it is used for raising awareness. Fifth, it is used for determining the effectiveness of water treatment technologies to find out whether properly water is treated or not according to the level required. Fifth, it is used for selecting an appropriate water resource. And also, at sixth and final last, that is to check whether the policies are sufficiently incorporated into the supply of water resources. So, in order to understand the water quality, we need to check it for the physical, chemical, and biological contamination. For that, we need to go for certain chemical tests. Physical water quality parameters are basically the water quality parameters that gives us the idea of solids, turbidity, color, taste and odor, and temperature. So these parameters constitute the physical water quality. Solids are classified into different types like settleable, suspended and dissolved solids. That is basically depending upon their size. Solids with a size more than 10 microns are called as settleable solids. Suspended solids constitute the particles of size 1 to 10 microns in diameter, and fine or dissolved solids are less than 10 to the power minus 3 microns in diameter. Suspended solids in water may come from different areas like inorganic particles, clay, silt, sand, glass, organic particles like plant fibers, algae, plankton, immiscible liquids like oil and grease. Organic solids are biologically degradable, but inorganic solids are not. There is significant psychological effects of the presence of solids that makes water aesthetically displeasing. Also, if suspended solids are present, there may be certain biological active compounds, organic compounds, that may cause diseases. Suspended solids provide absorption sites, and chemical and biological reagents tend to absorb into the surfaces, which further affect the treatment of water. Also, microorganisms that are present, they tend to reduce the efficiency of disinfection if suspended solids are there. But how do we measure it? For that, we need to adopt a gravimetric method. Gravimetric method is the one in which we basically weigh the sample and then we find it out with the help of calculations. For estimating total solids, we take a china dish, we weigh it first, we find out the weight of that empty china dish, then we keep a measured volume of the water sample in that china dish at approximately 100 to 110 degrees Celsius for at least the time for which the complete water evaporates. And after the evaporation of water, what is left out in it is the solids. So we again weigh the china dish after evaporating the water content and we find out the difference in the weight of that sample. That leads to the estimation of total solids into the given water. So if water has certain soil particles, it indicates there is some sort of contamination if excessive dissolved and suspended particles are present. Also, if water is clear, then it also impacts the rate of photosynthesis. For finding out suspended solids, we need to have a filtration assembly which is as displayed into the picture. We place a filter paper which is of a specific pore size of 1 microns in pore size. As we have already seen that the particles are classified according to their diameters, particle size, and those which are having particle size less than
10 microns are classified as suspended solids. So in order to find out suspended solids, it needs to be passed through a filter paper of that specific pore size. So this filter paper is having pore size of approximately 1 microns, through which the water sample is passed of a known volume. The suction pump is started so that the filtration can be done at a faster rate. Then, after passing the water sample, we remove this filter paper, we dry it into the oven, and after completely drying it, we again weigh it. So the difference in weight of these two samples, that is before passing the water and after passing the water, gives us the estimation of suspended solids into the water. Now we can find out the total solids content with the initial process which I mentioned. That is, we take the china dish, we pour a specific volume of water, we evaporate it, and then again we weigh it. Suspended solids we found out with the filtration assembly procedure. And when we subtract total solids from suspended solids, we subtract suspended solids from total solids, then we get the dissolved solid content. And in order to find out the fixed solid content, we keep that water sample which is evaporated into a muffle furnace at approximately 700 degrees Celsius for 15 to 20 minutes, at which the volatile solids escape out. We again weigh that sample, and finally the difference in weight gives us the volatile solids content. So when we subtract fixed solids content from total solids, we can get the volatile solid content. And settlable solids are found out by the help of Imhoff cone. So accordingly, we have discussed 5 different types of solids, that is total solids, fixed solids, volatile solids, suspended solids, and dissolved solids. For total solids, we have seen we need to have initial weight of empty china dish, then second, which is indicated in the form of W1. So W1 is the weight of empty china dish along with water sample. W2 is the weight of china dish in which we have already evaporated the water, that is the second weight. V over here is the volume of water that was taken into that china dish, and 1000 is the factor for which we need to convert it into milligrams per liter. Total fixed solids, after the initial procedure of total solids, this water sample which has been already evaporated is kept into a muffle furnace at a higher temperature, and the volatile content is evaporated. And then we find out the weight, which is indicated in the form of W3. This W3 minus W1, which is the weight of china dish along with the water sample, gives us this difference in weight, gives us the content in the form of total fixed solids. Now total volatile solids can be found out by subtracting fixed solids from total solids. And total suspended solids we have seen with the help of the filtration assembly. So W4 is the weight of filter paper before passing the water sample. W5 is the weight of filter paper after passing the sample. V is again the volume of water, and when we subtract total suspended solids from total solids, we get total dissolved solids. Let us discuss an example. So an analysis of determination of solids in the return sludge of activated sludge process was done, and it has following observation. A crucible was dried to a constant mass, which is 62.485 grams. 75 ml of a mixed water sample was taken in the crucible. The crucible with the water sample was dried to a constant mass of 65.020 grams in a drying oven at 104 degrees Celsius. The crucible with the dried sample was placed in a muffle furnace at 600 degrees Celsius for an hour. After cooling, the mass of the crucible with residues was 63.145 grams. We have to find out the concentration of organic solids in the return sludge. So as discussed earlier, this initial is W1, the crucible was dried to a constant mass, this, and 75 ml is the volume of water, so that is V. The crucible with the sample was dried at a constant mass, that is 65.020, and the crucible with the dried sample was placed in a muffle furnace at 600 degrees Celsius for an hour. After cooling again, its weight was taken. So in order to find out the organic solids fraction, what is important is that we need to identify which weight is to be taken. Should we take this crucible which was completely dried, or should we take it with the sample? So the crucible with the sample should be taken, and the crucible after completely drying and cooling, this weight should be taken. So this gives us the difference in weight from 65.020 and 63.14 grams. This difference in weight divided by the volume of sample, that is 75 ml, into 1000 will give us the amount of organic fractions, because in this case, after passing through the muffle furnace, the volatile section evaporates. So from calculating this, we need to get to know that the answer is 25,000 milligrams per liter, which you can check it into these calculations, as already I described this, that the weight of the sample of the organic fractions can be found out by subtracting 65.020, and this is 63.145 grams. We get 1.875 grams, and then finally we multiply it into 1000 divided by the volume of water. That gives out to be 25,000 milligrams per liter. We need to report it in the form of milligrams per liter. Moving on to the settleable solids, as already described, settleable solids can be found out with the help of Eimhoff cone. This, given in the picture, is the Eimhoff cone. It is of a specific capacity of 1 liter, in which water, which has to be tested for settleable solids, is kept for around 1 hour undisturbed into this cone. It has certain graduations into the bottom. After 1 hour of settling, the solids tend to settle into the bottom of the cone, and we can directly measure in the form of these graduations. It is reported in the form of ml per liter. The permissible and acceptable limits according to Indian standards for drinking water quality specify that total solids should be around 500 milligrams per liter, and if it goes beyond 2000 milligrams per liter, it is not acceptable for drinking purposes. Coming to the second parameter, physical parameter, that is turbidity. So to measure the turbidity, we need to have a specific instrument which is called as the turbidity meter. Turbidity meter is influenced by the term nephelometry. Nephelometry is basically the scattering of light when it is focused upon to the particles. Now turbidity is coming into the water samples because of the suspended particles. Suspended particles are fine particles which are not able to settle into the water sample and usually float, because of which there is a hazy, cloudy appearance into the water. So presence of suspended particles is usually not aesthetic. It also has psychological effects. We cannot drink contaminated water. Usually expressed in the form of NTU, that is nephelometric turbidity units, or ppm, that is parts per million or milligrams per liter. Certain impacts are there due to the suspended solids. It not only interferes with the penetration of sunlight into the water bodies, ultimately impacting the photosynthesis and the aquatic life. It can be measured by 3 methods. One is the turbidity rod or tape method, Jackson's turbidity meter, and the third is the Bailey's turbidity meter or nephelometer. Now turbidity rod or tape and Jackson's turbidity meter are already obsolete. Presently we are using nephelometer or turbidity meter for measuring turbidity into the water sample at lab scale. So this is the turbidity rod or tape method. in which there is a needle that is placed into with a scale. There is a scale and there is a needle placed into this along with that. It is inserted into the water body. Basically, it is the water which needs to be tested is taken into a jar and which is inserted. This tape is inserted into the water. We observe it from the top till the time when the needle goes invisible. That is, we are not able to observe this needle. And these graduations are then noted for finding out the turbidity. But now, this was a primitive method which was used to find out the turbidity to the water sample. Now it's an obsolete method. Jackson's turbidity meter, in which we used to have a candle at the bottom. We used to keep a candle at the bottom. We used to take a test tube in which water sample was filled in, and gradually we used to increase the volume of water till we are unable to see the candle's light from the bottom. Again, it was observed from the top, and finally the volume of water was noted to find out the turbidity. Again, this was a primitive method. Now it is not used. So 1 JTU, that is Jackson's turbidity units, is equivalent to 1 ppm, that is 1 parts per million. Both the methods of the turbidity tape or rod method and the Jackson's turbidity method, it depended upon the length of the path traveled by the light. So longer was the length of the path, smaller is the turbidity, and it is inversely proportional. Presently, we are using this Bailey's turbidity meter or nephelometer to find out the turbidity into the water sample at laboratory scale. This particular method is depending upon the principle of nephelometry. So, the first primitive methods which we discussed, they used to observe the light at 180 degrees from the light source. So, this was the light source and we used to observe it at 180 degrees. But when we change the observation part to 90 degrees and then we find out the scattering of the light, this is called as nephelometry or turbiditymetry, which is being used into the Bailey's nephelometer, also called as the lab scale turbidity meter. Now, acceptable limits of turbidity are 1 NTU. NTU is nephelometric turbidity units. And if it goes beyond 10 NTU, then we cannot use that water for drinking purposes. All the standards discussed here are according to Indian standards for drinking water quality, IS 10500-2012. Then we need to find out the color of the water sample. The true color is basically due to dissolved solids, and if there is presence of suspended solids along with dissolved solids, then we do not get a true color or we get an apparent color. There are certain physiological effects due to the presence of color. We cannot use it for drinking purposes. Obviously, you cannot drink a red colored water or green colored or blue colored water. Color basically indicates there is some contamination because potable water is usually colorless. We cannot also use these colored water for washing and drying. And certain chemical compounds, if their presence are there, they tend to react with them and generate color. Like, for example, if chlorine is present into the water, that reacts with certain chemicals and leads to the formation of color. How do we find out? We find it out with the help of a tintometer. Tintometer is basically an instrument in which there is a disc of a specified color, and then we compare the color developed with a standard solution. We insert it into this instrument and we compare it with the standard disc. So what is the chemical that's supposed to be added to have a color into the water sample? There is a standard solution of 1 milligram of platinum in the form of chloroplatinate ion that is dissolved, and then this solution is added to 1 liter of pure water. That leads to a formation of some colored water, which is compared with a standard disc as seen in the previous slide. But this testing should be done within 72 hours of collection of the water sample. Otherwise, the color is not the true color, it appears to be the apparent color. Now what are the limits? Acceptable limits is 5 TCU. TCU is true color units. And if it goes beyond 25 TCU, then we cannot accept that water for drinking purposes. Taste and odor indicates the presence of certain substance. Like if there is alkali, then in that case it causes bitter taste. Dissolved gases may also indicate the presence of certain contamination. Dead microorganisms, they also indicate the presence of certain taste. Mineral substances, carbonates and sulfates all change the taste of the water. So water has bad smell and odor and is objectionable if it does not has an appropriate taste and odor. How do we measure? We measure it with the help of an instrument called as osmoscope, in which the color and odor are given in the form of threshold odor number, which indicates basically the taste and odor of the water sample. Now the testing is done at normal room temperature because any change in temperature changes the biological activity. Threshold odor number is found out by the final volume of water after addition of pure water divided by the initial and original volume of water sample. That means we need to add pure water till we get a particular volume after the dilution and the initial volume. When we divide it, and we can find out the threshold odor number. This is done in an instrument called as osmoscope. If the threshold odor number is approximately 1, then there is no dilution required, and if it goes beyond 3, then we cannot accept it for drinking purposes. And the temperature. The normal temperature of any water body is approximately around 26 degrees Celsius. If it goes beyond 35 degrees Celsius, then it is not acceptable for drinking purposes because the change in temperature indicates that there is some contamination. If water is used, then in that case it leads to an increase in temperature, and used water is the only reason mostly which increases the temperature of any water body. So this table indicates the list of physical characteristics which we have discussed. The solids are estimated by gravimetric method. Permissible limits are 500 to 2000 milligrams per liter. Turbidity is done with the help of turbidity rod or tape and Jackson's turbidity meter along with Bailey's turbidity meter or nephelometer. The limits are 1 to 10 NTU. Color is found out by tintometer. The standard solution is 1 mg of platinum in the form of chloroplatinate in 1 liter of pure water, known as true color units. Permissible limits are 5 to 25 TCU. Taste and odor by osmoscope. Limits are 1 threshold odor number, does not need dilution, and if it's 3 threshold odor number, then we cannot accept it. And temperature, usually the desirable temperature is around 26 degrees Celsius. If the temperature of any water body goes beyond 35 degrees Celsius, then we cannot take it for water supply. And the change in temperature usually indicates that the water is used and then it is discarded. So this table gives a list of all the physical parameters and their permissible limits. Permissible limits are according to Indian standards, which are for drinking water quality, IS 10500-2012. So that's for today. We shall continue in the next lecture. Thank you.